Okay, well, let's get to the good stuff. So now I'm going to introduce you to Dr. Bohdan Khomtchouk, who is an instructor in the Department of Medicine at the University of Chicago. He actually spoke at our May seminar series and he introduced us to the field of cardioinformatics, which is the design, implementation, and application of large-scale bioinformatics and computational biology methods to cardiovascular disease, data storage, computing, and analysis. For this seminar, we have asked him to come back, and thank you so much for coming back to tell us all about your expertise. He's going to focus on his current research study and the work that he's currently doing with the Heart Bioportal. So we are going to go ahead and start the pre-recorded lecture, and then at the end, he's available to answer questions.
Renaissance, is it just me or is nothing playing?
It's not just you. We're having a little bit of a technical issue. And of course, this is what happens. We check it a million times. And then when it comes to the live show, we always run into something.
No worries. If the issue persists, I'm happy to give it live. So no issue whatsoever.
Give us one more minute. I'm sure we're going to get this. But thank you.
Hello, my name is Bohdan Khomtchouk. I'm a junior faculty member at the University of Chicago. I'm based in the Department of Medicine, specifically the section of Computational Biomedicine and Biomedical Data Science. I'm really excited to give this talk today about how to translate data into therapeutics for cardiovascular, renal, metabolic disease. The goal of this talk will be to show how you can create better drugs through bioinformatics and computational biology approaches, or what I like to call fighting heart disease with computation. The focus here will be on bringing together and integrating cardiovascular, cardiorenal, and cardiometabolic omics data. So by omics data, genomics, proteomics, etc., including imaging data, clinical records, biomedical literature, and other relevant data modalities. I'm happy to report that this project is now financially supported in part by the National Science Foundation, specifically their Innovation Core program, whose goal is to reduce the time and the risk associated with translating promising ideas and technologies from the laboratory to the marketplace. The I-Corps program is designed to support the commercialization of so-called deep technologies, or those that revolve around fundamental discoveries in science and engineering. So essentially, the I-Corps program addresses the skill and the kind of the knowledge gap associated with the transformation of basic research into what people call deep technology ventures. And I'm working with UChicago's Polsky Center for Entrepreneurship and Innovation this quarter to conduct primary market research around this project. So I'm excited to tell you all about all of this cool stuff today. So the disease focus area of this talk is on cardiovascular renal metabolic disease, or CVRM for short. And CVRM is the #1 cause of death in the world, and the market is huge. So here's a brief description. We'll get into this in later slides, but here's a quick segmentation into a number of different therapeutic areas. So you see, for instance, the cardiovascular disease market itself will grow to about 151 billion by 2025. The metabolic disorder market, which is composed of areas like type 2 diabetes, chronic kidney disease, etc., will grow to about 90 billion by 2025. So the market is absolutely huge. So of course the first step here, to translating data into therapeutics is breaking down data silos. So imagine how valuable a knowledge base this large can be, especially in machine learning settings as a training data set to power better, more predictive models. For example, one goal could be to build a specific scoring algorithm, let's say, that assesses a selection of targets for a specific CVRM disease and scores them in order of their potential to be best or first in class. So the scoring system can help reduce the selection process for users. Now, considering the many factors to evaluate when identifying opportunities for the next development of a first or best-in-class treatment, researchers are regularly challenged to rapidly evaluate the target landscape surrounding a specific indication or biological pathway. So ultimately, the primary goal is to support researchers with the process of selecting a target for the development of new drugs by providing the right context for decision making, which is what this is all about. So integrating multidimensional CVRM data into a single interactive search platform to support target identification decisions. Symbolically represented here as a cloud computing platform where you're putting together omics, images, literature, clinical records together, harmonizing that data, making it searchable and accessible for your specific target identification and prioritization needs. So now there's a lot of folks, right, out there, a lot of folks interested in this market segment. And part of our NSF requirements are actually to perform customer discovery interviews with research and early development teams at these above listed companies. So as you can see, there's a lot of representation here across both big pharma and biotech. And of course, technology like this would also be useful to preclinical academic research, not just industry. But speaking of industry, right? So earlier this year, my colleagues at University of Chicago helped Bristol Myers Squibb, BMS, as they're more commonly referred to in abbreviated form. They helped BMS build their own internal data infrastructure platform to house their internal proprietary data sets. So BMS is actually one of the biggest big pharma companies that's directly involved in cardiovascular, renal, metabolic disease drug discovery. So this is a particularly exciting piece of news. And then this summer, BioIT World awarded the Innovative Practices Award for this, actually, to my colleagues at UChicago. So the keywords here are, let's read it real quick. So the ability for researchers to find, access, and share genomics data in order to maximize the potential of data-driven science is still a large challenge for pharmaceutical organizations. So that's sort of the key part here, is that it's still a large challenge for pharmaceutical organizations to bring together, consolidate, curate, and harmonize, and make searchable and accessible all of their internal proprietary data sets, both clinical data sets and genetic data sets and other types of data sets that are specifically a part of their drug discovery programs and their early R&D teams. So taken together, right, this of course presents an untapped market opportunity. So one way that we're capitalizing on it is by creating biotech spin-off companies from my lab. So for example, Nucardia. So one of these companies is called Nucardia. So this is a screenshot from our website, www.nucardia.com. And as you can see, we're in stealth mode at the moment. And our goal is to serve the data analytics and infrastructure needs of biotech and pharmaceuticals industry clients engaged in cardiovascular, renal, metabolic drug discovery based on proprietary technology originally developed at Stanford University and University of Chicago. So Stanford University was where I was previously based. As a postdoctoral research fellow, I was an American Heart Association postdoctoral research fellow at Stanford prior to taking a faculty position here at the University of Chicago. So speaking of market size, like we mentioned, like right at the beginning, right, the market size is pretty substantial. So if you look at, for example, the metabolic disorder market alone, it's almost $90 billion, where the rising incidences of diabetes, obesity, and hypercholesterolemia are expected to drive the market growth in this sector over the next 5 years. And our goal is to create a knowledge base of cardiovascular, renal, and metabolic disease information that's specifically optimized towards target identification and prioritization. And our new platform will hopefully reduce the potential for target identification failure, greatly improving the chance of developing a first-in-class or best-in-class CVRM drug. Also, the end user market for drug discovery contract research organizations, otherwise known as CROs, is actually expected to reach 85.8 billion by 2022, rising at a CAGR, compound annual growth rate of 9.4%. It's not something that I listed out here specifically in the slide, but I need to add that bullet point next time I give this talk. Okay, so next up, so let's talk about unmet need. As you might imagine, right? So selecting a druggable target for preclinical development it's actually really hard, especially in cardiovascular, renal, metabolic therapeutics pipelines that have very complex biology. So, cardiovascular, renal, metabolic diseases, very, very complex, right? It's very systemic, right? There's a number of comorbidities. So a person who presents with heart failure may also simultaneously have type 2 diabetes and chronic kidney disease. Many of these diseases also have endophenotypes. It's just very complex. Selecting the right target is arguably the most important step in the entire drug discovery process. And of course, taking the wrong first step in the selection process is very costly, both time and money wise. You can spend up to a decade and sometimes more chasing basically after a ghost and wasting a lot of time and money. And on average, it's like depends what kind of drug you're developing, but between $1 and $3 billion is what it costs for 10 to 12 years of development in this space. It's very, very expensive. Sometimes it goes up to like 10 to 15 years in this space for a single drug. So picking the right target is just really, really critical. Also, building and scaling up your own infrastructure to support data-driven drug discovery decision-making is a huge investment, as you might imagine, right? So think of it this way, right? Take any biotech company or any big pharmaceutical company. Think of their bioinformatics and computational biology teams and what they do on a day-to-day basis. In terms of analyzing data. A lot of that work is not directly relevant to actually scaling up infrastructure, which is a very software engineering kind of based task. So it's actually it's something that companies internally do not necessarily have. Their computational teams don't necessarily specialize in those areas. So it's a really difficult challenge and requires a lot of time to create infrastructure like this. And internal R&D it's a cost, right? It's not an asset. So speaking of Nucardia, the idea behind Nucardia is to mitigate risk and associated expense for clients by, for example, developing and scaling up the technical infrastructure platform for you, so that you don't have to, right? So you can spend time doing data analysis and other tasks that are mission critical to your drug discovery program. Also helping you bring together and harmonize cardiovascular, renal, metabolic disease information across, at scale, across multiple heterogeneous data modalities and repositories. We're talking both in-house proprietary data sets as well as any kind of public data sets that you've personally manually curated, harmonized, and made available as a part of your early stage R&D systematic target identification and prioritization. And also then applying, cutting edge AI machine learning, statistics, bioinformatics, computational biology methods on this big data to enable better predictive modeling for systematic CVRM target identification and prioritization. So in a nutshell, right? So speaking of this biotech spinoff that my lab was working on, we want to accelerate the time to discovery and reduce the potential for target identification failure. Once again, greatly improving the chance of developing a first-in-class or best-in-class CVRM drug if this is the therapeutic area that your enterprise is focused on. So our focus is on helping organizations build and scale an internal precision medicine platform to enable this kind of drug discovery based on our unique skills and expertise developing Heart Bioportal and Gen 3. So in the rest of this talk, we'll focus a lot of our time on Heart Bioportal, but big shout out to Gen 3, a technology upon which Heart Bioportal is built. It's sort of the technology that you use to build data commons. So really encourage you to check it out. I think the website is gen3.org, and it's a phenomenal resource. So it's also open source technology that you can use to scale up data commons. And this is what Bristol Myers Squibb was using to scale up their first sort of internal genomic data commons that we saw a couple of slides ago, and for which BioIT World awarded the Innovative Practices Award. So Nucardia can then help create powerful AI machine learning algorithms based, like basically think of this as a target deconvolution algorithm, right? You're looking for how to systematically, identify, targets and prioritize targets based on the clinical indication that you're targeting, and other aspects. So, what if you could, create based on this infrastructure, target deconvolution algorithms trained on data sets in this platform, and then augment them as needed with public data? It's a huge value add, and Nucardia contributes significant value add there. So in terms of creating your internal database infrastructure, plus offering an extra sort of layer or service of data analytics services and expertise within this space. And the goal, of course, is to facilitate making better CVRM drugs through large-scale data analysis and computing in the context of a digital drug discovery dashboard for your specific enterprise. Some short and long-term objectives. So what we hope to accomplish in terms of translational objectives over the next couple of years, with help from the University of Chicago as well as the National Science Foundation, is we want to figure out what regulatory landscape, for example, this kind of computational technology would come across. Also, we want to find potential commercial partners to conduct pilot studies and ink strategic collaboration agreements. That's really important to, get the ball rolling and start, with the discovery phase. I think that that's pretty much what we're going to be exclusively focused on the next couple of months. Also, integrating traditional drug mining techniques. So, for example, high throughput screening of drug libraries with our technology. Determining a service offering that fits well within existing drug discovery pipelines, including, of course, making key introductions to key individuals who can help determine product formation milestones. Navigation of the complexity associated with intellectual property and university licensing, right? Considering that this work was started at Stanford University while I was a AHA a postdoctoral fellow, and then taking that work with me to the University of Chicago and continuing it here, and now spinning it off from my lab. And then, of course, as a potential exit strategy, we could sell the tech and the platform to a big pharma company at some point, or identify CVRM-specific drug targets that can be channeled through the university's technology transfer licensing office. Some other players in the market. This is something, right, that sort of you have to say it first, because you have to understand how big this market is, right? Like, this is the number one cause of death in the world, and there's so many comorbidities, as I mentioned. It's very systemic, very complex biology domain. So there's a lot of opportunities for innovation in this space. Examples, for example, of previous blockbuster drugs in this market include Bristol Myers Squibb Eliquis, so Apaxaban, and Johnson & Johnson Xarelto. So speaking of Eliquis, interesting story. So my grandfather, he was actually one of the very first patients to receive Eliquis, and he had deep vein thrombosis. So he had really bad, thrombi. So his entire bottom left leg below the knee was like blue. Like, it was literally blue. So he was signed up, and this was many, many years ago. His cardiologist signed him up as one of the first kind of patients to receive Eliquis, and today, like, his leg looks absolutely fine, and he actually credits his life to Eliquis, and Eliquis is a phenomenal drug, personally, I think, because it pretty much saved my grandpa's life. So very indebted to the drug discovery innovation that's been coming out of BMS. It's just absolutely phenomenal. I think Eliquis, though, is coming off patent within the next couple of years, so it's going to be a generic. So not quite sure what the next steps there are, but very, very interesting territory. So Eliquis, it's a prescription medicine used to reduce the risk of stroke and blood clots in people who have atrial fibrillation, which is a type of irregular heartbeat. Similar to Eliquis, Xarelto from Johnson & Johnson is used to treat and help prevent blood clots related to a specific condition like deep vein thrombosis or a pulmonary embolism. Also, you have examples of newly emerging drugs. For example, inclisiran is intended for patients with hypercholesterolemia. to reduce their LDL cholesterol levels, while, for instance, mavacamten, or MAVA, is used to treat symptomatic obstructive hypertrophic cardiomyopathy. Notably, by the way, Myocardia, the company that developed mavacamten, they employ whole exome sequencing to create targeted therapies that work best in patients with specific genomic profiles. So they're like the quintessential sort of knowledge company that's developing a precision medicine platform to stratify using genomics their patients for their clinical trials. Patient subpopulations stratified using, bioinformatics data analysis approaches, is what they work on, with a focus on hypertrophic and dilated cardiomyopathy, including heart failure. That's from my understanding of myocardia, or at least, from what I've read in the press releases, and I follow the company pretty closely, and very excited about some of the amazing innovation that's been coming out since it was initially founded in 2012. Really, really cool company. There's a critical unmet need for more and improved precision medicine approaches, like the one that Myocardia took to discovering mavacamten, and I think that computational efforts to enable and sustain such innovation are just now really emerging in terms of like both recognition from a marketplace perspective as well as from an academic research perspective, and also in terms of other companies coming into this space. So, and plus a lot of it, is being developed in my lab, and that's a lot of the technology that we're spinning off now into Newcardia. So I've always been driven to create computational innovations, that have real world impact. And actually, the organizers of this seminar series have asked me to give some of my background and career trajectory. So I have a brief slide here to go through that. So basically, long story short, I've been at UChicago for almost 2 years now. During this time, I've focused in on developing tools and software to bring together cardiovascular, renal, cardiometabolic data from various databases. And I'm working on initiating a field called Informatics. So that's the last bullet point here with footnote with superscript 3, footnote 3. So basically, I'm trying to bring computation to the field of cardiovascular, renal, metabolic disease at a very large level. So trying to get people excited about entering this space. So when you're a bioinformatician or you're a computational biologist, just pick any random bioinformatician or computational biologist that you personally know. If you ask them what do you work on, most of the time you'll hear that, yeah, I'm working on something cancer related, or I'm working on something in the neurodegenerative disease space. But you will almost never, or very rarely hear, cardiovascular disease, or, renal diseses like chronic kidney disease, or, cardiometabolic diseases like type 1 or type 2 diabetes, or the more interesting pre-diabetes now. You'll almost never hear that. And I want to change that culture, and I want to put spotlight on the fact how important it is to do that, right? So, like the kind of impact that precision medicine has had on oncology has been tremendous in terms of bridging the not only the bench to bedside divide, but also the informatics to medicine divide that still very much exists in biomedical data science. So. Looking at that is absolutely fascinating, and I think it's a really important area given that cardiovascular, renal, metabolic disease is the #1 cause of death in the world. So I'm a junior faculty here, as I mentioned, right, at UChicago. I've created a platform called Heart Bio Portal over the last 3 years or so, slightly over 3 years now, maybe even 4 years. No, like three. It's the world's largest precision medicine platform for cardiovascular disease genetics and genomics data. As I mentioned, I was a former AHA postdoctoral fellow at Stanford. I was based in the Department of Biology and the Department of Medicine. During my time at Stanford, I was also a research associate at the VA Palo Alto healthcare system, where I worked on cardiovascular genomics analyses in over 450,000 at the time, by the way. They have way more now. But at the time, it was 450,000 whole genome sequences from Korean and Vietnam War veterans enrolled in the Million Veteran Program. Currently, I'm a super user with administrative access to over 300,000 cardiovascular, renal, and metabolic disease relevant human whole genome and whole exome sequencing data sets. in NHLBI's biodata catalyst ecosystem. And my primary expertise, of course, is in CVRM disease biology. So as I mentioned, I initiated the field of cardioinformatics. I have a Ph.D. in human genetics and genomics with dissertation papers published in journals like, AHA journals like the Journal of the American Heart Association, et cetera, et cetera. I did my postdoctoral training in cardiovascular medicine while I was at Stanford, and I'm currently a PI, a bioinformatics PI at UChicago. And there's a link if you wanted to explore, additional details. So research mission-wise, I want to build a world-class research program in the emerging field of cardioinformatics, working at the nexus of bioinformatics and precision cardiology, perform service and public outreach, and actively pioneering and promoting cardioinformatics to the scientific community. The goal is to fight heart disease with computation through the creation of large-scale heart data commons technology like Heart Bioportal. I also advocate for the need to, as I mentioned, not only bridge the bench-to-bedside divide, but also the informatics-to-medicine divide in cardiovascular healthcare. So an example of, what does it mean to fight heart disease computation? What does that actually mean? So as we've been discussing in this talk, right, data-driven CVRM drug discovery, so systematic target identification and prioritization, using various kinds of omics data in Heart Bioportal. So Heart Bioportal has a ton of different omics data. It's got genomics data, proteomics data, genetic association data, gene expression data, a ton of different data modalities, and it's only growing. So I'm an active developer and maintainer of the system. So we're always trying to add more data and more data modalities to make cardiovascular disease data and cardiorenal and cardiometabolic data more accessible to researchers interested in this space. And the ultimate goal, right, of course, is to impact healthcare and with it, genetic medicine. So actually, one fun thought that I have in the back of my mind is maybe one day to launch an American Heart Association journal like Circulation Cardioinformatics. That could be really cool to bring a recognition to the fact that we need more people, the new generation of bioinformaticians and computational biologists that are graduating from for instance, the MSc in Biomedical Informatics program here at UChicago. For them to get excited about applying their skills and their knowledge to fight heart disease with computation, to make a dent in this space. And the space is wide open, huge market opportunities and huge room for impact. And some recent work from our lab. So as I mentioned on a previous slide, right, we have a lot of stuff cooking, a lot that I can't really discuss on a public talk like this, but we will go through a lot of really interesting things that I can discuss and things that we actually have bioRxiv preprints out on. That's currently, papers that are under review at various journals. So, for example, we've been talking about Heart Bioportal, right? It's the world's biggest human cardiovascular and cerebrovascular disease genetics genomics data platform. So HBP, Heart Bioportal 2.0 will be published sometime next year. And there's a preprint out for it, by the way. I'll show you, I think, in one of the next slides. But you can Google it. You can Google around for it, and you'll be able to read it. So the first paper was actually published in Circulation, Genomic, and Precision Medicine, and then the second paper we sent to Database, which is it's an Oxford University Press journal. So recently we as this bullet point says, we engineered new computational drug discovery knowledge graph features to enable data-driven systematic target identification and prioritization. This specific development has not been added to the bioRxiv preprint that's currently in review. That's something that we're going to be holding on to until version 3.0 and probably projected for early release in 2021. But we'll talk about that on the next slides. So right now, it's currently in private beta. So public release is scheduled for next year. And here we go. So here's the two papers. So on the left is our research letter that we sent to CircGPM. So that was published in April of 2019. And now, actually, when did I submit this? October 21st. So October 21st, I put out a preprint in bioRxiv, and you can check it out as well. It's an expansion of Heart Bioportal into various new areas with a focus on ethnically diverse human populations that are afflicted by cardiovascular disease. Really, really cool updates in that paper. So if you're Heart Bioportal user, definitely encourage you to check it out. But speaking of the knowledge graph features for drug discovery, so actually back in August, we tweeted out our intentions to enter the space. So we said, if you're a drug hunter interested in cardiovascular disease, we'd love to get your pre-release feedback on Heart Bioportal's upcoming features. We'll reach out individually to drug hunters and cardiometabolism experts who enter their emails here, and then a link to a MailChimp URL. By the way, if there's one kind of like thing that I would really suggest is always talk to users. So, when you're developing data commons and you're developing new features and you're adding new data sets, like you really want to address an unmet need. And you want to talk to your users, especially your power users, and ask them, what's lacking, what needs to be added, what would they most love to see. So, for example, like we have a Survey Monkey out that you can take. So if you go to Heart Bioportal, there's like a link in the menu that says take our survey. So you can like take the survey for Heart Bioportal and say what you like, what you don't like, rate, what you think about different aspects of the platform, etc. So that plus reaching out to individual users who have registered to the system and just, sending out an email asking, if there's anything that, that they would most love to see in upcoming releases of Heart Bioportal. And we learned a lot in this process in terms of especially of, what would drug hunters love to see. Very, very insightful comments and suggestions, and we're really excited to roll these features out publicly. As I mentioned, they're currently in private beta. And regarding that MailChimp link, so for example, this is what it looks like, right? Basically, if you're willing, it says to offer your advice, please enter your email address, and we will reach out to you individually. So we got some really great feedback using these channels. And here's another screenshot, right, where we provided some information about us, some pictures of some diagrams, some figures and charts that you can see when you search for various gene names and see what's known about, like gene X and disease phenotype Y. And these are the sort of plots that sort of consolidate all that information and make it visually user-friendly and easy to digest. And also, then you can press data download buttons that'll actually pull down like a nice and tidy CSV data files, like flat files that give you all the information that you need to do additional sort of follow-up analyses on or secondary analyses of the work that we've done. So the product roadmap for us, our focus is going to be on building a precision medicine platform to continue building a precision medicine platform to enable cardiovascular, renal, metabolic drug discovery. And the goal, as I mentioned, is to facilitate making better CVRM drugs through large-scale data analysis and computing. And we want to, as I said, we want to bring together large-scale genomics, proteomics, metabolomics, et cetera, basically omics data, including CVRM-relevant imaging data, clinical records, biomedical literature, et cetera, and sort of create the world's most powerful precision medicine platform for CVRM disease. So build a digital drug discovery dashboard and let the user have the power to do large-scale data analytics on data of that size. And when we talk about size, currently Heart Bioportal has several terabytes of data behind it. So it's a really large system. I mean, it's not petabytes, like for instance, the Genomic Data Commons, which is the most advanced cancer data ecosystem built on Gen 3 technology. It has petabytes of data. So we're not there yet. And there's a number of reasons why we're not there yet. For example, it's a lot more common in the oncology community to release your data sets, so make them open source, make the summary statistics available if you're doing GWAS, or make the the FASTQ files available, if you're doing next generation sequencing. That's really not the case so much in the cardiovascular disease research community. In fact, there's a lot more data hoarding that goes on in the in the cardiovascular, renal, metabolic disease data community in terms of data. So it's a huge problem. It's actually, we'll probably give a talk just on that alone. So that's definitely one of the unmet needs of what needs to change in the culture of how we do research in this space. And maybe that's something that, for instance, the NIH's NHLBI can delve into more and sort of change their policies on how things are done, and what incentives are provided to researchers to not data hoard and all these other things. But I don't want to get off topic, go off on a tangent on that. Here's a quote that was said by our chief of the section of cardiology at UChicago. So he said that the field of cardiovascular disease has always been data and technology driven, but when it comes to taking advantage of the vast amount of information that already exists and developing a systematic approach for analyzing it, the field has fallen short. And the emergence of computational biology and bioinformatics in the cardiovascular field will provide insights and potential therapeutic avenues for the treatment of cardiovascular patients. So here you see a press release that came out in December of last year called Research Sets Sites on Heart Disease Discoveries through Precision Cardiology and Bioinformatics. And definitely encourage you to check it out if you have time. Regarding progress to date, so HeartBio Portal drug discovery features now include, and this is in private beta, so it'll be rolled out sometime next year. We have 550 druggable targets, so genes, gene names, and respective drugs. We also have 182 cardiovascular and cerebrovascular disease phenotypes, and to date we've collated. So let's see. Let me see if I can recall. So okay, so HeartBio Portal users can now search for any potential gene target to obtain things like, for example, target name, target class, target tractability, druggability, biological pathways, related diseases, what else? Corresponding drugs and their relevant details, of course, including clinical trial status of the respective drugs, its protein binding data, biological half-life, bioavailability, mechanism of action, all those things, including toxicity, efficacy, pharmacodynamics, metabolism, etc. So all these results are presented graphically with tidy data downloads available directly from the HeartBio Portal user interface. And the user interface looks like this. So here's what it looks like. So basically, the HeartBio Portal interface, it enables the user to look up a gene of interest. So in this case, for instance, right, you're looking at gene TNNT2, it says at the top. And then have drugs relevant to that gene, as well as key characteristics of that drug, like I mentioned, regarding bioavailability, mechanism of action, et cetera, be loaded and ranked, right? So it's a search algorithm, essentially. So searching for a gene name will initially return information about the top drug hit for that gene, toxicity and efficacy data, and also an overview of the number of compounds across clinical trial phases associated with the target gene. So that's panel 1 here. Clicking the See All Drugs button in this last set of data actually reveals an expanded list. So you'll see like pharmacodynamic data and other information that's specified for each hit. That's panel 2. And if multiple stereoisomers are found for a specific formulation, so a notification is then labeled forms and stereoisomers, and that offers the ability to click through to an interface that offers data on the first firm to actually gain approval. So the first company to actually gain approval for a given stereoisomer, as well as the year of the first marketing approval for each enantiomer. So that also includes available literature on the different effects of each enantiomer. Each enantiomer's data then also contains information on the country of the first approval and whether the enantiomer is available as a generic drug, as well as a structural visualization. So that's in panel 3. Regarding how we put all this together, so regarding methods, so what we did in order to get the data powering, the user interface in the last slide, here's what we did. So first we queried, right? We queried all the human-related genes in Ensemble, for instance, and found there, by the way, and other resources, including like Ensemble's not the only one. We didn't want this, number 1 bullet point to be really long. And find their corresponding sort of accession identifiers through a combination of querying the BioThings API. So mostly we're using mygene.info and myvariant.info. Then we find the related diseases and drugs of these genes using the Open Targets API, and then filtering those genes that are specifically related to CVRM phenotypes. Then we search, curate, and harmonize each respective drug in databases like Unichem, PubChem, Combol, Reactome, etc., to present actionable information most useful for biomarker discovery and validation efforts. That includes, as I mentioned, biological half-life, toxicity efficacy, pathways, networks, as well as other pharmacodynamics and protein binding drug information. And that includes the stereoisomers of the drug molecules, if available from the aforementioned databases. So that's to assist biomarker-guided drug repurposing efforts from pharmacogenomic data. And then we concatenate all that CVRM relevant data together and produce clean JSON files that record each gene protein target with its associated clinical indications, drugs, and actionable metadata useful to drug hunters searching HeartBio portal. So by providing gene and variant level search, essentially across various CVRM related data modalities, so omics, text, etc., we mined and extracted existing and hope to predict novel target disease associations to assist early drug target decisions and biomarker discovery efforts, including indication prioritization to facilitate safe and effective development of the next generation of cardiovascular, renal, and metabolic therapeutics. We're sort of going almost at the limit of the time. Acknowledgements here. Gratefully acknowledge Stanford Spark Drug Development and Translational Research Program for early discussions from 2018 to 2019, which is where I was based prior to coming here to UChicago. And then here at UChicago, I'm really grateful for pilot award funding from the Center for Translational Data Science, and also grateful to the National Science Foundation I-Corps program for funding through the UChicago Polsky Center. So with that, I'd like to open it up to questions, and thank you all very much for coming to my talk. Thank you.
Thank you so much. Like Will said, we will open it up for questions, so you can just put them in the chat box and we'll read them out. All right, looks like we have a question. Okay, I am Norbert Nowenko. I hold a PhD in bioinformatics from the De Montfort University. I've been involved in translating genomic information, protein sequences, to biological functionalities using a Fourier transform-based digital signal processing techniques. Where am I? Oh, including resonant recognition method. This technique will be of relevance here. Apart from our 1,000 products analyzed by the Khomtchouk et al. And Khomtchouk, I've been able to invent biomedical devices, including computer-aided drug-resistant calculator. Let me know if you're interested. Okay, well, we have some interest for people to work with you, Dr. Khomtchouk, so that's great. Do we have any questions from anyone? Anything you want to ask?
Yeah, I just wanted to add to Norbert to connect with me offline if he'd like to talk further about this. And he can reach out to me with my email address. So my email is my first name, Bohdan @uchicago.edu. Happy to get his email. There's another question coming in here. For those who are not looking for an MSc degree, will any classes of the program be offered on an open enrollment basis? That's probably a better question for Renaissance McIntyre and Christine Clark, since they run the program.
Hi, yes. If you are not interested in actually earning the degree, you can apply through the Graduate Student at Large program, and through that program, you can take courses in the BMI program. So while we're waiting on questions to pop up, what I want to actually do is just provide a little bit of information for anyone that is here that is a prospective student. Kelsey Diaz is our assistant director of enrollment management, and she can provide you with information, about, for the person that asked about just taking courses. She can provide you with information about the Graduate Student at Large program, so you can just, take a select few courses that you're interested in. or if you're interested in earning the degree, she can also provide you with information about how to apply for the program. So I'm going to leave that up, and then, I'm going to see if we have any other questions, and we can get those answered.
Also, I just wanted to shout out that I am also active on social media. So I'm fairly active on Twitter. My handle is Bohdanome, Bohdanome, like genome, but my name and then OME at the end. I typically cover a lot of topics within this space. So I frequently either like or comment or retweet on topics within the cardiovascular, renal, metabolic drug discovery field. So if you're interested in sort of keeping up to date with what is interesting to me and what I'm following and sort of what I'm reading, you're happy to connect with me and follow me on Twitter. Also, I'm active on LinkedIn, so you can find me on there as well. And yeah, so and then also you can always shoot me an email to my bohdan@uchicago.edu address. Happy to hear from you.
Thank you so much. We are going to keep the call for just a couple of more minutes. We do have students that are in this session, and they do have class tonight, so we don't want to keep everybody for too much longer, but we will stay open for a few more minutes, and then we'll end the session.
And thank you for everyone for coming as well. I know it's 5 p.m. here, Central Date Time, so I'm not sure where everyone's calling in from, but it is, later in the day, so I appreciate everyone coming out at a later hour.
It was great. Thank you so much, Bo.
Thank you.
All right, well, we're getting a lot of thank yous in the chat box. So I think people are probably winding down for the evening. So we're going to end it here. Thank you so much for coming and, giving us an update on the new research that you're working on. It's very, very fascinating. So thank you. Thank you so much. We are going to provide a recording of this to the registrants and for the students that are on the Zoom call tonight. This link is also going to be in the Canvas site. So I want to thank everybody for coming out. Thank you so much to Bo for coming and talking about his research. Thank you to Jarvis and Chris for all the work with putting this together. And our next seminar series is not going to be until January, but we'll send information out about that. So have a good evening, everyone, and thank you.